Support for this grassroots community television program comes from U.S. Trust. From wealth structuring to investment management, U.S. Trust's global perspective, unique team approach, fiduciary platform, and more than 200 years of experience provide for the kind of insights, solutions, and expertise that have a worth all their own.
Good evening. Hi. I want to welcome you all to this evening's lecture. This is part of the Aspen Center for Physics Heinz Pagel Summer Public Lecture Series. Before I introduce tonight's speaker, I wanted to say just a few words about the Aspen Center for Physics for those of you who are not familiar with it. It's a gem well-known among theoretical physicists. Of course, it's here in Aspen, which is a gem in its own right. But the center brings together something like 500 of the world's leading physicists every summer, each physicist staying for a few weeks to participate in one or more programs that focus activity on new topics, old topics that still need to be solved. And it's just a wonderful tradition among the physics community for people to see each other that they don't have another chance to meet, to start new projects, to solve old problems. One thing that is also useful or seminal at the center is that it builds communities among groups who don't otherwise have a place to meet. So for example, tonight you're going to hear a talk about biology. Well, there are a group of theoretical physicists and some experimental physicists as well who've taken a great interest in biology, but there's no perfect meeting place for them. There's no society of which they're members. And the Aspen Center for Physics has been a wonderful place to bring these people together, to meet each other, to cross-fertilize, and that's happening right now. So there's a program going on right now. So a few more things that I want to say before I introduce the speaker. Use the microphone? Okay, I'm sorry. I'm being recorded apparently for posterity. There will be lectures throughout the summer. The next lecture will be a week from now, June 19th. Robert Kirshner from Harvard will be giving a lecture called The Accelerating Universe, Einstein's Blunder Undone. Apparently the only mistake Einstein ever made was to think he had made a mistake. So you can find out about that next week. But this week, the topic is going to be something a little different, not from the conventional repertoire of work done by physicists. It will be a talk focusing on problems in biology. And the speaker tonight will be Casey Huang, a good friend and colleague of mine. I'll tell you a few things about him. He has a very distinguished pedigree. He was an undergraduate at Caltech, graduate student at MIT, a postdoc at Princeton, where I had the pleasure of working with him, and he's now a professor at Stanford, and somewhere in there he spent a year in Cambridge, although he tells me he really just had a good time that year. Something that's not apparent from those facts is that Casey has made a transition. His early training was in pure physics, and somewhere down the road he realized that there were interesting problems for physicists to think about in biology, and he's fully transitioned to become a practicing biologist. His lab at Stanford has all the sorts of things that you would imagine in a biology lab, but he still brings his special physics perspective, asking the kind of questions that physicists ask and demanding the kind of rigor that physicists demand. And so he will tell you this evening his perspective on a bug's life with the same creativity, rigor, and charm that we always expect from Casey. So here he is.
Thanks very much, Ned. Can everyone hear me? Yes. Great. I think Ned has a special bias for distinguished pedigree since I followed him to Caltech, MIT, and Princeton. So it's really a pleasure to be here today. Thank you very much for the wonderful introduction, Ned. And it's really a special privilege to be able to tell a group of interested people like yourselves about our science. As Ned mentioned, I've been trained as a physicist, which means that I absolutely expect interruptions and questions during the talk, so do not disappoint. As Ned said, I've been trained as a physicist, but I've found something very compelling about biology and the set of questions and problems that it gives. I did realize after the fact that my choice of title might leave some misconceptions about what to expect today. So this is a scene from A Bug's Life, where Flick here is tasked with finding ways to fight off the invading grasshoppers. I will not mention grasshoppers for the rest of today. You may be familiar with the fact that bugs in computer programming can take on a life of their own. This is a famous page from a notebook from the Mark I computer, where I've zoomed in here to the moth that was trapped in one of the relays. And it was not actually how the term bug was coined, but it's a nice urban legend. So this is also not the topic of my talk, although we do have a lot of bugs of our own in our code. This is actually the bugs I'm talking about. So these are soft toys of different bacteria that you can find in nature, some good, some bad. And my son has a few of these, in particular this one, because it's my favorite bug. This is E. coli. And I'm going to tell you a lot about E. coli, but hopefully give you some sense of how we're trying to understand the general rules that apply to E. coli and to all bacteria that you find in nature. So this is an electron microscopy image of E. coli cells. It's false colored. E. coli is rather boring colors in real life. But it has what I think of as a very beautiful shape. So you can see that one cell is just this long rod. And you may find this boring, but I think this stirs up an amazing number of physical questions about these cells. For instance, as you can see here, all these cells seem to be roughly the same size. They seem to grow and divide in a particular direction. Somehow they find the middle of their cell to divide. They maintain the diameter of the cell as they elongate. All these questions somehow beckon to physics in the sense that this can't be the action of one protein, one gene. It's really somehow a collective action that the cells take on. And I have to say, when I was working with Ned, I came to these sorts of problems assuming that these were questions that had already been answered. That was, of course, naivete, because after working on them for 10 years, we still haven't solved many of them. But I am going to tell you the answers to some of them today. So the first thing I want to impress upon you is that bacteria really are everywhere. And you may be familiar with some of the ways that bacteria impact your life. One way is simply through the biogeochemical cycling that goes on throughout nature. You can see all over this chart that bacteria are important for nitrogen fixation. They're important as decomposers. In fact, decomposition occurs not just in the soil and in plants, but it occurs in organisms as well. So this is a Hawaiian bobtail squid. And the first time I came to Aspen in 2005, Bonnie Bassler from Princeton gave the public lecture about this amazing organism that has a symbiotic relationship with bacteria called Vibrio fischeri that live in the gut of this squid and produce light. And that light is actually used by the squid in order to evade predators. So this sort of glow that you see is actually being produced by the bacteria themselves. Now, bacteria are, of course, also found all over the human body. The Human Microbiome Project is an enormous NIH-funded project to study the diversity of bacterial species that you find all over the human body in many different contexts. And to put it in perspective, every human has about 100 trillion cells in their body, and about 90 trillion of them are bacteria. And so one way that people have started to think about this is that we are just the puppets and the bacteria are running the show. Now, bacteria in your body affect an enormous amount of processes, some good, like the ways that you process nutrients that you consume in your food, how your immune system matures, your metabolism. It's even been shown, at least in mice, that bacteria can control the sleep patterns of the mice. This last part may or may not be good, depending on your perspective. There's certainly bad things that bacteria are known to affect, like diabetes, obesity. But actually, bad is probably a bad connotation here, because if you'd like to think of this as a balance between you and your bacteria, then really I think what a lot of these signify is that somehow that balance has gone out of whack. This last one I highlight because something that's been in the news a lot recently is so-called fecal transplants that are incredibly effective against things like inflammatory bowel disease. If you don't know what fecal transplants are, you can figure it out just by thinking yourself. You know, that belies the fact that most of the bacteria that you find across the human body are where else? In your intestines. So the thing I'd like you to think about is that there are 90 trillion bacterial cells, roughly speaking, in your intestines, but this randomly chosen human being here on the right, 2 years, 8 months, and 15 days ago, actually was completely sterile. And so interestingly, humans, other mammals, are born sterile and acquire their so-called microbiota as they transit out of their mothers. And this was actually his first visit to the dentist, where he was getting some of his microbiota removed. But fortunately, this incredibly complex system can be studied in a variety of different ways. And that germ-free human can be recapitulated in the context of a germ-free mouse. And so scientists over the last 20 years have figured out how to take mice, remove all the bacteria from their bodies, and then propagate the mouse colony in that way in these incubators right here. And so I've actually done a few of these experiments myself. It's incredibly stressful because you don't want to get even one cell onto these mice. But what you can do is to interact with the mice in various different ways, give them whatever bacteria you want, study how that community evolves. And I'll come back to that later on in the talk. But what this has enabled is the idea that we can take mice and turn them into something like the flasks of bacteria that we grow in the lab in order to have a very controlled environment, or at least an environment that we can control as well as possible. So E. coli is one of the bacteria that you can find in your gut. Can be good or bad. But it's, of course, one of many thousands, if not millions, of species of bacteria. And one of the things that I find compelling, in the same way that I found compelling about looking at E. coli, was just to see the different shapes and sizes that bacteria take on. Actually, this one right here is the favorite of every graduate student that passes through my lab. It's called Stella humosa. It forms these 6-pointed stars. And this is actually a cell in the process of dividing. You can see that it replicates its shape incredibly well, just like the E. coli cells. This is a bacterium that forms many different stalks. These stalks are thought to be used to attach to surfaces, which is a very important part of the bacterial life cycle, both out in the wild and inside a host. This is a bacterium that lives in freshwater called Colobacter, which is a model organism for differentiation of cells because it actually forms 2 different cell types. You can see this one here sticks to surfaces, this one swims away, and there's a very exquisite program that takes place in order to encode those 2 different cell types. This is a bacterium called Thiomargarita, but I want you to notice here this scale bar here. So this is a millimeter here, this white bar. And so these enormous bacteria are single-celled microorganisms. We don't know very much about them, but I think this shows how incredibly diverse the bacterial kingdom can be, where you can go from an E. coli cell up to something about 10 to 12 orders of magnitude larger in volume. This is a spirochete, which I just love because this very regular pattern seems to be important not only for how they move, but also how they invade cells inside your body and things like that. Yeah, question?
All those green circles are singular cells?
These are all single cells. Yes, thank you. This is a chain of single cells. As you might expect, there's huge problems associated with how you communicate from one side to the other. And in fact, this is almost like a thin spherical shell where there's a huge vacuole in the center and then tens of thousands of copies of its genome spread throughout this very thin cytoplasm around the surface of the sphere. It's an interesting organism, although the subject of almost no work except to make pretty pictures like this. So having seen this incredible diversity of shape and size, what I hopefully will convince you over the next 10 minutes or so is that morphogenesis, the dynamics of how cells determine and maintain their shape, is a dream problem for a physicist like me. It's really one where I think it not only requires the sorts of tools that physicists have generated in order to deal with inanimate problems, but also conjures up new problems that I think can feed back on the way we apply our tools to physics problems. So the reason I think it's a dream problem is because despite all that diversity, it can be reduced down to a relatively minimal set of components. So bacteria, like all cells, have a membrane that surrounds their cytoplasm. So this is what divides the cell from the external environment. But also around them, just like plants, they have a very rigid cell wall. Okay? So the cell wall covers the entire membrane. And the very interesting part is that the cell wall is both necessary and sufficient for how cells get their shape. So here's the evidence for that. So we can use enzymes to strip away the cell wall. Okay, so lysozyme is something your cells use to kill bacteria. The reason it kills bacteria is because it makes the cells very sensitive. A lot of the cells will die, but if we're very careful not to let them die, then we strip away the cell wall. What happens is that the cells take on these round morphologies. So each one of these black circles is a spherical cell. It's lost its shape completely. Actually, this is an E. coli cell that no longer has its wall. So that's demonstrating that the cell wall is necessary for cell shape determination. But we can actually do the exact opposite and take away everything but the cell wall. We can use enzymes that digest the DNA, the RNA, the proteins, the membranes, and what we end up with is one huge macromolecule. It's one molecule that sticks together, and this isolated cell wall has the same shape as the cell that it came from, thereby demonstrating that the cell wall is sufficient for cell shape determination. So this again is E. coli, has this rod shape, but even if you take one of those six-pointed stars, the cell wall itself has that same shape. So I think that illustrates the fact that that translates the problem into understanding what is the program that leads to each one of these specific cell shapes. So I think I wrote something in the abstract about a cosmological view of biology. I think that's somewhat grandiose, but I do think that we face the same sort of problems, whereby we know a lot about the proteins that exist inside the cell. They are on the size of about a nanometer, or a few nanometers. The cell itself is a micron or larger, and somehow those tiny proteins have to be able to detect and alter this much larger cell shape. Okay? And so we'd like to understand what are the rules that allow the cell to translate across these different orders of magnitude of scale. So one thing I haven't talked to you about is growth. So let's think about growth for a second. If I start with a single bacterial cell, let's say it's an E. coli cell. After one generation, you get 2, and then 4, and after G generations, I have 2 to the G cells. This is exponential growth, right? These cells divide by binary fission, and so this is the picture that you probably have in your mind of how bacteria grow, just like other cell types. But let's do a simple estimate using arithmetic. So this is the area of the Earth, and this is the area of an E. coli cell. Hugely different. But that allows us to calculate that it would take about 10 to the 26 E. coli cells to cover the surface of the Earth. Okay? So E. coli cells double every 20 minutes. So how many generations would it take to make this many E. coli cells? Well, some simple arithmetic tells you that 2 to the 90th is about 10 to the 27th. So 90 generations would be required, and 90 generations would take about 30 hours of growth. There's, of course, enormous flaws with this argument because this would also take probably something the size of the Earth in terms of nutrients to feed all of these E. coli cells. But I think this belies the point that E. coli cells, just like every other organism on the face of the Earth, are not always in this phase of very fast growth. And in fact, these sorts of studies date back to the 1940s, where Jacques Monod actually classified the different phases of growth. And this is going to become very important later on in the talk. So what I'm showing you on the top is the log of bacterial density. So you can think of this as just how many cells exist. And on the bottom here is the growth rate. So it's just the derivative or the rate of change of this function. So you can see everywhere that this function is flat, the growth rate is 0, here and here. And you can divide this up very roughly into phases. So there's what's called a lag phase, where the cells haven't quite gotten going in growth. It's sort of like when you get up in the morning, it takes you a while to get going. But once they get going, they really do have a period where they double exponentially. They're undergoing this exponential growth where their growth rate is constant. Now, what you might imagine happening is that the cells exhaust all of their nutrients. They also produce a lot of waste products, just like us, that their sister cells don't like. And so that leads to what's called stationary phase, where the cells either stop growing or even start to die later on. Okay? And so what you can see here is that these three discrete phases of growth may have very different impacts on how we think about how all of the bacteria on our body, in the rest of the earth, are proliferating and occupying the different niches that they live in. So one of the most powerful aspects of bacteria is that they have been evolving for an incredibly long time, billions of years, and they have occupied many different niches. But it's, of course, very difficult to understand 2 billion years of evolution in sort of the span of a couple decades of a scientific career. And so Rich Lenski, who's at Michigan State University, took upon himself to kind of recapitulate evolution, but in a context that is not nearly so complex. So what he did in 1988 was to inoculate 12 cultures of E. coli. Starting from the same culture, each one of these flasks got just a little bit of his original culture. And this is some text from his website where he says the inexorable rhythm of the project is as follows. Basically what he does is every day he takes a little bit out of each one of these flasks and puts it into a new flask. So it's going through the cycle where it grows up and then exhausts its nutrients and then goes into a new flask so it can do that day after day after day. He's been doing this since 1988, so he's gotten about 60,000 generations, and every 500 generations, he saves this. This is like the fossil record that allows him to go back and ask what sorts of changes in these cells were responsible for their different behavior. So one of the powerful aspects of this is that we can very quantitatively measure fitness. So we can ask, how have the cells changed over time? And so this is the sorts of measurements I'm going to be showing you, where you mix the ancestor with some evolved strain. I'm going to call it a mutant, but somehow it has some changes that have occurred. And if we mix it at a 50-50 ratio so that we get a well-mixed culture of both green and red, making yellow, then we can ask, after 24 hours of growth, which allows for about 7 generations, given how much nutrients there are, we can say, OK, are there more green cells or more red cells after all of this? So let's say there are more red cells. In fact, let's say there's about 95% more red cells. Well, 1.95 is about 1.1 to the 7th power, which means we can sort of backtrack out to say there's about 10% fitness gain per generation as these cells are growing. Now, this, of course, is a very coarse description of what's happened because, as I mentioned, these cells are going through a very complex cycle where they get going in the morning, they double for some period, they go into stationary phase, they might die. But like I said, this is sort of a standard way of thinking about what it means to be better in the context of evolution. So one of the things that you might not be surprised about, given the principle of natural selection, is that cells get more fit over time. So this is the first 10,000 generations of his experiment. And you can see that if you sample every 500 generations, the cells are getting more and more fit in competition with the ancestor. This is not so surprising. What is surprising is that amazing things can happen in these experiments. So for instance, this is the number of cells that you get out after a day's worth of growth. And you can see that it's pretty flat until about 33,000 generations, and there's a massive jump. And what happened is that these cells learned to metabolize citrate, which is a molecule in the nutrient source that the cells could not digest before. So they've literally just learned a new way of eating, and this enabled them to grow to about 5 times higher cell count at the end of the day. I think this is a particularly interesting aspect of, again, trying to replay the evolutionary clock in ways that might tell us new things about how cells function. So I think this is just one opportunity to drive home the point that bacterial evolution is something that's occurring all the time. It can be incredibly fast. This is a somewhat morbid excerpt from Alexander Fleming's Nobel speech in 1945. I will let you read it. Basically, he's saying, let's imagine that Mr. X goes and gets some penicillin, doesn't really pay attention to how big the dose is, and he doesn't take enough to kill all the bacteria, what he's going to do is produce bacteria that can live with the penicillin, and then he can pass this on to his wife, who then dies, and then where do you place the blame? This wasn't much of a pick-me-up, but I think this illustrates the point that this is something that impacts our everyday lives when we think about the fact that evolution can occur on timescales that don't involve billions of years. So here's the question I wanted to ask. These cells are getting fitter. That's not surprising, but are they changing physically in any way? And I'll just tell you the answer is yes. So these cells are actually growing faster over time. So I would like, as a show of hands, how many people in the audience think that they get bigger as they get more fit? And how many people think that they get smaller? Okay. So, I mean, of course, this is a trick question in some ways. Almost everyone answers smaller because it just makes more sense. You're dividing faster. Let's just do that when you're smaller. And the amazing thing is that they get much bigger. So over those 10,000 generations, the cell volume doubled. So they are not only growing faster, they're also growing more every time they do so. Now, I find this incredibly intriguing because a doubling of cell volume can impact just about everything. Now, I think from a biological standpoint, you could view that as sort of incredibly complex, but I thought perhaps we could apply some tools from physics to try to understand what is the basis for this cell size change and what impact does it have on the physiology of this organism. I have to say, the problem is that the molecular basis of how cell size can change is virtually unknown. So we're really starting with somewhat of a black box.
A question?
Yeah.
That curve was so smooth, there were no fluctuations, whereas your curve of fitness, they were all over the place, statistical fluctuations. What's behind that?
That's a great question. I have to say, if I were a reviewer of this paper, I might ask for some error bars on these measurements. This would, so I think it's a fascinating difference, and as I'll come to in a few slides, I think what this stimulates is a question of, is bigger better? If so, what does that really mean from the context of the cells? And is one being more tightly regulated than the other? I'm not going to be able to answer that question in response to this data, but I will tell you that I think we have a much better handle on that bigger versus better relationship, and I'll show you that. So this is work done by a postdoc in my lab, Russell Mons, and he came at this with a very broad conceptual framework where he wanted to say, okay, can we achieve some synergy between this experimental evolution paradigm and cell biology? So can we have evolution teach us about what cells are doing now, and perhaps in the reverse as well? So he started from a very similar experiment. The details are not particularly important, but I'm going to show you some data from this specific experiment where we took the same ancestor that Rich Lenski started with and just evolved it in 4 different media now. So every day, it either got glucose as a food source, lactose, both of them, or one day glucose and one day lactose. This was to study something completely different, but what we were able to ask is, has cell shape changed over the course of evolution? So after about 2,000 generations, we did the simplest thing we could, which is just to say, okay, all of these evolved strains, how do they look under the microscope? And these blue diamonds are the ancestor in each case. They are all in the lower left-hand corner, meaning they are the smallest. And all of the evolved strains are in different colors, here, 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 and here, and they are bigger. This is in agreement with what I showed you before. But this particular one really caught our eye. It's much bigger than the rest of the cells. In particular, it's much wider than all the other cells. It's gone up in cell width by about 30%, and it has almost a 2-fold change in cell volume. So it turns out that that strain has 7 mutations, but 6 of them are not important. So this evolved strain, as I told you, it's about 30% wider. And it has a mutation in a gene that I'm going to tell you about in a second called MREB that just simply takes an alanine amino acid and substitutes a threonine. That's it. And the cool part about this is that that particular mutation is necessary and sufficient for changing cell width. So we can take this ancestor and just introduce this mutation, we get wide cells. We can take this evolved strain and switch the mutation back to the alanine that it was before, and the cells go back to being skinny. Okay? So this is a single mutation that confers this very large change in cell size. So what is this protein doing? Yeah?
Did the mutation depend on what specifically it was fed?
Great question. And I'm going to come back to that in a few slides. But I will just give away the punchline to say that this mutation makes the cells much more fit depending on what it's being fed. And I think that's very fascinating, but it also gives us a beautiful physical handle for deconstructing what causes the cells to be better or not. So this protein MREB is really the only protein I'm going to talk about today, and the details are not important, but we were particularly interested because it is already known to be very important for how cells grow. So this is a crystal structure of an MREB protein, and it forms filaments. So this is one MREB and a second MREB, and this 53rd residue, which has an alanine in normal MREB, is right at the interface between these two proteins. Already kind of interesting, since these polymers are actually very structurally similar to a protein called actin that you're found in all of your cells. It's one of, if not the most abundant proteins inside your body, and it's a so-called cytoskeletal protein, which participates in an enormous number of processes. But as you can see in this fibroblast, these actin filaments stained here in green seem to give the cell its shape. And in fact, they are very important for how cells grow. And in bacteria, the same thing is true. So on the left here, what I'm showing you is MREB proteins tagged with a fluorescent protein so that we can see where they are. And they're actually very dynamic. So this is the time here on the left. And what they're doing is rotating around the cell. And they're doing so as the cell grows. So what we can do is here on the right, we can add an antibiotic that stops cell growth. And that stops this rotation. So you can see here that the pattern is just static. Okay? So through this and many other experiments, what we think is that this protein is actively directing where the cell should grow. So it's very intriguing to think that a mutation can change how the cell grows as well. But, yes?
Are you suggesting that just getting larger confers an advantage?
I'm not suggesting that yet, but I'm going to show you that it does. And so I think that's the question that we wanted to answer, because nothing I have shown you has really told you about causality. And in fact, what this requires is a new set of tools that we've generated in order to specifically address that question. But first, is it ever better? So this is not going to say that bigger is always better, but is it ever better? And the answer is yes. So what we did was we took every carbon source we could get our hands on, and we grew these cells, either the ancestor or the mutant in each one of these carbon sources. What's interesting is if you change the food source for these bacteria, you change the cell size. But also, in every one of these food sources, this mutation makes the cells wider. Okay? So no matter what you feed it, it's going to get wider. So does it get more fit? So just like I told you in these competitive fitness assays where we can ask for something like a 10% fitness advantage based on the ratio after they grow up, what you can see here is that for many, many carbon sources, the mutant is actually something like 10% more fit. That's an enormous advantage. Keep in mind, that means every day there's twofold more of the mutant than there was of the ancestor. Very quickly, the ancestors are going to be gone. However, that's not always true. So glucose is a sugar that bacteria really like, but lactose is as well. Lactose has no fitness advantage whatsoever. So we can propagate this for days and days and days. The mutant and the ancestor just sit there in happy equilibrium with each other. So coming back to the question before, we think this is a powerful tool, even though we don't understand what makes glucose better than lactose. Because anything we postulate for making the cells more fit has to agree with this fact that there is a difference between these two sugars. So, coming back to this question, are bigger cells better? Well, we have a single mutation that causes cells to be bigger and causes them to be more fit, but that does not mean we can draw an arrow like this. So Russell, in his, yeah?
2 slides back, what's interesting is the one that he less fed.
Right. In galactose, yeah, the cells are actually penalized for being bigger. And I must admit, I have no idea why that's the case. Galactose is an interesting sugar, but there's nothing particular about its chemistry that would tell us why this is. So in Russell's very first presentation to our lab, he drew this cartoon. And he said, wouldn't it be fantastic if we could really controllably change this axis and then measure what happens along this axis, and of course, wouldn't it be our lucky day if there was something really smooth and systematic that occurs? And because he's a very clever guy, he actually figured out how to do this. So what he did was he said, okay, if this alanine to threonine substitution makes the cells wider, what happens if I take this alanine and change it to something else? So there are 20 amino acids, 19 besides alanine, and a large fraction of those make E. coli into a rod, but a rod with very different cell widths. So you can see here that the cells get fatter and fatter and fatter as you introduce a number of different mutations. And so I'd argue that this is about as systematic a way as you can change cell size. And on the right here, the cell volume is about 2.5 times larger. So this is the data that he sent me one day with a subject line in the email, "I promise I did not make this up." And as you can see, there's really this beautiful linear relationship where as the cells get bigger, they are getting more and more fit. So this is all in glucose, whereas you can see here, the A53T has about a 10% fitness advantage. We find this very powerful because what it means is that any hypothesis that you apply to these cells to argue why these cells are better has to apply in a very continuous way across the spectrum of cell sizes. So why are the cells better? Remember that I told you that the cells, yeah. It is getting shorter, and that's a very interesting aspect. It is not getting shorter nearly enough to make the volume go down. In fact, the volume goes up quite a lot here. The reason, one way to think of why the volume is really going up quite a lot is because the volume scales with the width squared, whereas the length is just contributing as a single power. But these cells are over twice as large in volume as these cells here. Yeah. So what's actually happening? Well, remember I told you that cells go through this period where first they're not really growing, then they accelerate up into exponential growth, and then they level off. In this case, they're not really dying when they hit stationary phase. But as you can see, if we just grow the mutant and the ancestor, they actually have very similar growth rates during this period where they're doubling exponentially. And in fact, if we grow the ancestor and the mutant on the exact same surface and just watch them over time, so you can see 0, 25, 50 minutes, what you can see is that the mutant cells, which are fatter, highlighted with these red arrows, actually seem to just start growing faster. And if we just measure the growth rate of the mutant versus the ancestor, they reach the same growth rate, but the mutant just gets there faster. It's like it just gets the head start that it needs to occupy all of the space, and after that, they just grow at the same rate. So this is particularly interesting because, like I told you, if I want to hypothesize that some general mechanism for getting more fit, then it has to apply across all of these different cases. So let's think about lactose. Remember I told you in lactose there's no fitness advantage. And indeed, in lactose, you still see that the cells get wider as they do based on the mutation. But you can see here that the growth rate is exactly the same for the ancestor and the mutant. And so what we think this means is that there really is no acceleration into very fast growth in lactose for reasons we don't understand, but that was the head start that it got in glucose. And that really explains the difference between these two food sources. But going even further, just like I told you, we've got these wide range of cell sizes. Does this apply across this entire range? And it does. So all of these cells are growing at basically the same growth rate, maybe 1% of variability between these different strains. But the lag time relative to the ancestor, meaning how rapid of a head start does it get, just goes down, 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 down. And then once you get to the part where fitness plateaued, basically this lag time advantage just becomes the same. So this is really interesting to us because what it means is that if you want to think about why cells are evolving to become fitter, maybe you don't want to think about how they actually grow. Maybe you want to think about how they get out of not growing. Okay? So the other question this conjures is, okay, well, why aren't all bacteria huge? And coming back to the point at the beginning, maybe this is about just them not being able to settle into quite the right environment, but maybe there's some trade-off. So this is an experiment done by Natalie Balaban. And while I start this, let me just talk you through what's going on here. So this is in a microfluidic cell where you can watch cells grow. So these E. coli cells have been, are expressing a fluorescent protein, and then an antibiotic called ampicillin killed almost all of the cells, except for these two. What you can see here is that this cell basically just sat around and waited but didn't die. And after a while, once the antibiotic was washed out, this cell was able to start growing again. This is not a mutation, because if you add the antibiotic, all these cells are going to die again. This is what's called a persister. It's a cell that can stick around in the presence of antibiotic, but doesn't have any sort of genetic advantage relative to its sister cells. Now, this is incredibly important from a medical standpoint, because what I'm showing you here is a macrophage that's been infected with salmonella cells. This is before antibiotic treatment. And these two cells have been labeled with one fluorescent protein. The rest here have been labeled with a different fluorescent protein. I won't go into the details of how this was actually achieved, but actually these two red cells are persisters. And after antibiotics, they survive in the cell, whereas all the rest are dead. And one of the really interesting aspects about this is that these cells are basically not growing. They're not going into this rapid phase of growth, which is what allows them to survive the antibiotics. And one of the things that has emerged in the last year or so is that these persisters across a wide range of species are much smaller than their sister cells and have these much longer lag times that allow them to sort of sit in this dormant state. So perhaps the fact that cells are not big in nature is somehow related to the fact that there's a benefit to staying small, sitting tight, and waiting for those harmful situations where you need to survive and everyone else is going to die. That's one possibility, and this question mark should, of course, be taken with a lot of importance because, well, maybe the cells are bigger. So this is a random sampling of strains from the Broad collection at MIT that collected environmental isolates of E. coli. And interestingly, every environmental isolate that we looked at is bigger than the strains that we see in the lab. I don't really know what that means, but I think it does belie the fact that what we need to do is study not just E. coli, but all organisms in as close to their natural environment as we can. So I'm going to take a very slight detour because I think this is a very important aspect, not just of my lab, but how science is evolving and how physics really is contributing to a rising wave, I think, of data in biology. So the NIH has a huge initiative called the Big Data to Knowledge initiative. And I think there are a lot of people in the community that wonder how big data actually gets translated into knowledge and doesn't just stay as ones and zeros. And part of the challenge here is to train people that can think about the biological knowledge and how to manipulate these big data sets. So how big are data sets? Well, YouTube actually gets about 100 hours of new video per hour. 100 hours of new video per minute uploaded. This translates to about 10 petabytes per year, which was the sum total of all digital information on the planet in 1986. So there is an incredible rise of digital information. What is this useful for? Well, physicists have found a lot of interesting applications. So this is a paper that was published where what they did was to take movies from YouTube of mosh pits at heavy metal concerts. And just they developed some code to analyze where all the people were going. You can see here their trajectories. And then they were able to fit this to theoretical models of flocking and group motility. And the data fits the model incredibly well. You can get all sorts of interesting patterns like vortices inside these mosh pits. They also note in the acknowledgments that no federal funding was spent on heavy metal concert tickets to do this research. And I think perhaps this is the tip of the iceberg. I think maybe data can be used in ways that the people who are gathering the data don't even expect. But I think this involves actually training the people who are going to be looking at the data to look at it in new and interesting ways. So this is a class I teach at Stanford called Computational Modeling of Microbial Communities with Tiffany Vora, who's here in the audience today, and Justin Sonnenberg, who runs a germ-free mouse facility at Stanford. And the goal here is to train the next generation of biologists to think about data in the way that we do as physicists, where we try to reduce it into simple models that can be used to make predictions to guide the next round of experiments. And we're sort of the philosophy that this is a learn by doing field, where we really can't just teach you concepts. You have to apply them to a problem that you care about. And so every set of students gets a project that has an unknown answer and is asked to, over the course of 10 weeks, come up with some research. And one of the projects that we underwent for the first iteration of this course was to study the spatial organization of the mouse gut. So just like I told you, germ-free mice start out with no bacteria whatsoever. But what you can do is to take a germ-free mouse and inoculate it just like you do a test tube with whatever you want. We could inoculate it with a single microorganism. We could inoculate it with human feces and ask how those populations evolve inside the mouse. We do this for about 10 days. We can in particular control what sort of food the mouse is getting. What I'm going to show you is results from rich and poor dietary polysaccharides. And then what we can do is to sacrifice the mouse, collect the tissue, and in this project what we want to do is see where the bacteria are in relation to the human cells, how many of them are there, and can we understand something about their organization. So we took this data and gave it to the students and said, tell me what's there. And this has turned into an ongoing project in Justin and I's lab because the results were so compelling. So what I'm showing you here is a slice through the colon of a mouse. And this is many, many images that have been stitched together. So it's about a 300 megapixel image. And what you can see here are three different labels. So in green is an antibody labeling the mucus that lines all of the epithelial cells in the gut. In blue is a DAPI stain that stains the DNA of all of the cells in the so-called lumen that's outside of the human cells. And we can do this in both a standard diet or in a diet that's lacking polysaccharides. What I hope you can notice here on the right is that if we take a small section of this shown here in this white box, that the mucus is actually much more spread out when you have a very rich diet. This is particularly interesting because it's known that bacteria, when they get starved, actually will start to eat the mucus. What we're seeing here is we think, a very direct signature that if you don't feed the bacteria, they will eat you. And I think it has very interesting implications for how this balance between the host and the bacteria will adjust to changes in things like nutritional patterns. So this was, again, that very rich food source that the mouse was getting. And what you can see here is now a very high-resolution zoomed image where here is the boundary of the epithelium. In green, again, is the mucus. And in red here, what you can see are individual bacterial cells. Shown here on the right, what we can do is just to computationally extract this boundary, stretch it out along a line, and ask how close are the bacteria coming to this mucus, potentially to start eating it away. And the answer is that they're coming very close. But also, what we can see is now that there's a very widespread of pattern of behaviors in these two environments. So you can see here with standard diets, there's a much wider range of thickness of the mucus. It gets much more tight and skinny when you don't feed the mouse and the bacteria this rich diet. So what I think this belies is the fact that one needs this sort of rich data set, but one needs to also come to a very simple conclusion through computational analysis that can embrace this big data and reduce it down into a much simpler picture. So coming back to what I started with, one of the questions you may be asking yourself is, if all of this complexity exists in the mouse, where the bacteria are not just growing, but they're interacting with the host cells, does what we get in the laboratory when we evolve these bacteria actually tell us anything about the challenges that bacteria face? So this is something that we would like to answer. And so I'm not going to tell you the answer because, honestly, I don't know the answer. But I would like to just crowdsource this problem where what we have done is to take the ancestor and the mutant and combine them into a mouse and ask who wins. Maybe if I'm sufficiently compelled in the question and answer, I'll tell you what I think I know. But how many people think the mutant will win? And how many people think the ancestor will win? about 50-50. I think, I think this is sort of the future of my lab, and perhaps I would like to think is the direction microbiology is going, where we can make incredibly systematic experiments in the lab that allow us to very subtly perturb what's going on inside the cell, what's going on outside the cell, really ask what are the general rules that allow these cells to grow and proliferate, but also embrace the fact that biology is very complex. These cells have had to deal with an enormous variety of circumstances, and particularly with this very powerful approach of using germ-free mice as a sort of incubator of cells in the same way that we may be incubators of the bacteria in our body, ask how many of these things translate into these sort of real-life environments. So I just wanted to highlight Russell Vaughn's, who did all the work in my lab on cell size and how it relates to fitness. I also wanted to highlight some of the imaging work done by Tim Lee, and a fantastic set of graduate students and postdocs, which I think probably the most interesting thing for this audience is that I will just label them as bioengineer, physicist, bioengineer, geneticist, structural biologist, biophysicist, biochemist, and evolutionary biologist. And I think merging all these things together has been incredibly fun for me. I hope it's been the same for you to hear about it. I wanted to thank collaborators Tim Cooper and Miguel de Pedro for the work on the evolution and fitness, funding sources, and in particularly Aspen Center for Physics. It's been a real pleasure to talk with you today, but also, I come every summer to Aspen. It's an incredibly invigorating environment, both at the Physics Center and across the town. And now my son has been here twice, and I think he's hoping to be here for many more summers. So thank you very much. Yeah, it's a great question. And I think we can do this across a very broad range. We have not, and those sorts of questions have not even been posed in any experimental context. I think, it's not surprising, given that it took about 30 years for Rich Lenski to get to where he is today with these evolution experiments, that no other organisms have been studied in this context. And so one does not want to get too focused on one particular organism. But I think now what we can do is exactly as you say. Go in, ask how can we perturb different organisms, and ask how they evolve. And so hopefully we'll be able to figure that out. Yes?
Going back to where the man killed his wife.
Yeah. Yeah.
Was it, if he had finished the dosage of antibiotics,
Right.
did I get that right?
Yeah. So we use antibiotics as an incredibly powerful tool for genetics because they will kill the cells. And so what we can do is to, for instance, modify the genome in such a way that only the ones we want will survive when we give antibiotics. But, the fact of the matter is that it takes about 2 days for us to evolve a strain of bacteria that is resistant to a particular antibiotic. So basically, almost every new antibiotic that is generated these days, within a year certainly, there are clinical isolates that are resistant to that antibiotic. It's one of the rising issues in especially the third world, but it's not just that, based on overuse of antibiotics. It's estimated that something, I think, like a third of antibiotic prescriptions are basically just given out because the doctors don't want to argue with the patients. In fact, a third of people surveyed think that antibiotics are the best way to treat the cold, which of course is a virus and does not respond at all to antibiotics. So I will say that I don't know the primary data behind this, but what I've read is that it is much better to stop a dose early rather than take partial doses for a long time. One of the reasons why is because of what Alexander Fleming alluded to, which is that if you just give a low dose, the bacteria kind of get used to it They figure out how to live with this antibiotic, and then you've got a big problem on your hands because the bacteria then can learn to tolerate more and more and more, and that's one way people think is the sort of pathway to these very widespread antibiotic-resistant strains. Yeah?
Yes, this is a general question.
Mm-hmm.
As a non-scientist, you began this lecture
Mm-hmm.
on the E. coli strain.
Right.
Are these studies applicable to other strains, other viral strains?
Yeah. It's a great question. So I think one thing you could ask is, and I think what we'd like to ask is even more broadly, what is the relationship between how cells grow and their shape and size? And so, for instance, cell shape has been linked to a lot of different changes in cell shape have been linked to a lot of cancer cell lines. Shape and size can affect things like the differentiation of stem cells. I think in each one of those cases, what you should be asking yourself is how much of that is actual causal relationships and how much of that is just, well, you change one thing, lots of things can change in a very indirect way. So I think the power of bacteria is that we can manipulate them very precisely in the way that I showed you to try and test those sorts of causal links. But I have to say, it could be that everything I've just told you is specific to E. coli. I think not, based on the fact that the substrate that we use to change, which is this one protein MREB, is very well conserved across a wide variety of different bacteria like Pseudomonas, Salmonella. And in each one of those cases, it appears to do the same thing. So I wouldn't be surprised to find that the effects of perturbing it are the same. But I have to say that what you've highlighted is one of, I think, the next great challenges that my lab and many other labs end up focusing on one organism. I think we really need to kind of link all of them together in order to be able to ascertain whether we've got rules or we've got some sort of specific interesting incidences.
Actually, that brings to mind for me how you define that fitness is not the ability of an individual bacteria to be relational,
Mm-hmm.
to form symbiotic relationships with other organisms to create larger organisms,
Mm-hmm.
to further ensure longevity. If you look at how we are now, it's the
Mm-hmm.
it's the combination of many bacteria to create what we think is like a single organism like a human.
Mm-hmm.
So isn't that a better way to define fitness?
I think that's definitely a very interesting and different way to define it, and I think those sorts of questions have to be asked as well. They're of course harder to ask because, for instance, in the case of E. coli, one doesn't know what it really cares about when it's inside the body. There are thousands of different bacteria. They're all participating in different chemical reactions. But I think this sort of germ-free mouse paradigm, where one can build from the ground up, is a way to start tackling that. Yeah.
How difficult is it to stain a particular protein that you specify?
Yeah.
In this case,
It's easy to do and difficult to interpret in the following way. So I won't jump back. A protein's a few nanometers. A fluorescent protein, also a few nanometers. And so if I ask you to give someone a piggyback ride, it will change your behavior. You might be able to go just as fast. I cannot. And so that is effectively what you're asking the protein to do when we label them. And so it's an incredibly powerful tool. It was awarded the Nobel Prize 2 years ago for the discovery of green fluorescent protein and its application in biology. But I think it's one of those challenging questions. Like, how can we observe really at the nanometer scale what's going on inside the cell, but not change what the cell is doing? What I've showed you in these fluorescent protein movies with MREB is a system where it does not have its endogenous MREB protein. It only has this MREB with the fluorescent protein, and it dies without MREB. And so we think it's basically doing the same thing because the cells grow, they look the same, they grow at the same rate. But it always has to be in your back of your mind. It's one of those aspects of biological tools where even the most powerful things can go wrong sometimes. And so I think what one has to have also is many complementary tools to start coming at it in different directions.
In a particular case, you have to have a different set of
I think what you have to have is a very precise definition of what it means for the protein to work.
Okay.
And so in this case, we do. And so it's a very useful way to ask, did we break anything by introducing this fluorescent protein? And since we think not, we can interpret the activity.
Okay. So that's the part that takes a very long time.
That's right.
The rate when from the parent to
Mhm.
the next
Right.
generation,
Mhm.
what do I want to say?
As you're propagating the cells, or as just as one parent produces two daughter cells?
Well, when you have your miss reproduction,
Mm-hmm.
how does that rate compare to other cell
Yeah.
breakdowns in other organisms or other cell structures?
If I understood you correctly, how does this sort of production of daughter cells really vary either in one organism or?
For the I can't think of the word right now. For the when you've got a mess up in the
Yeah.
transcription of the
Right.
cell.
Yeah, that's
The word I'm trying.
that's a complex question because there's many different ways the cell can go wrong. But I think one of the fascinating aspects of bacteria is that they can live in many, many different environments that will change how fast they can grow, but they still do so with this incredible fidelity that really there isn't a lot of breakdown except in extreme circumstances where, for instance, we are trying to kill the cells with antibiotics. And in many other environments, really, you can define what growth means in one particular environment, move it to another environment, and do the same. Although I have to say, I think it's fair to say no one knows. When I put a bacterial cell into a host, no one knows what actually happens. Do they just divide like crazy? All we know is what comes out the other end, because that's what we measure. And so I think the next step to a lot of this imaging work, where we try to ascertain where the cells are, is where they're going. And so what we're trying to do is actually image inside the mouse gut while the mouse is alive. It's not an easy task, but I think it can be done. Yeah?
You have debris labeled in yellow. What exactly is that?
Yeah, those are actually polysaccharide material. One of the interesting aspects is that a huge fraction of about 10% of the calories that you use for energy come from food that you are not metabolizing, but the bacteria are. In fact, in mother's breast milk, a third of the sugars that the mother synthesizes are completely useless to the baby, but they are specifically utilized by the bacteria that colonize the baby. And so, in fact, one thing you might notice is that these very large pieces of plant material are almost absent in this case, and this is the case where we didn't give it very much food. Yes? Yeah, okay. So we did a 15-day experiment, and sometimes the mutant won, and sometimes it lost, but it never lost completely. It stably settled into this coexistence where either it was about 90% or it was about 10%, depending on the mouse. And so what we have done now is to start putting in 90% mutant or 10% mutant and ask what happens. I have to say, it's not what I expected, not even necessarily what I hoped for, but now I'm really fascinated because I think the fact that you can get this divergent behavior either means that, it could mean that it's just noisy, but I find that unlikely given that it really settles down into a stable environment. Or what it really could mean is that, there's something about the way that the bacteria come in or something about the host itself. And so I think perhaps this is a controlled environment where we can start to figure that out.
But these mice are all genetically the same, wait for a long time.
They are genetically the same in one sense, although I have to say, working with bacteria shields you from some of the realities of life. For instance, when we started the second experiment, the mouse facility technician asked, do you want boy mice or girl mice? And I was like, oh, I never realized they come in 2 flavors. And so I had to go back and say, do we even know what we started with in the last experiment? Turns out we started with all boys, and so we're now we are actually locked into only using boys in case boys and girls behave differently. So, you know, I think that's an interesting question, but they're basically all coming from the same litter, is what we try to shoot for. Yeah. Okay. All right. Thanks very much.
Support for this grassroots community television program comes from U.S. Trust. From wealth structuring to investment management, U.S. Trust's global perspective, unique team approach, fiduciary platform, and more than 200 years of experience provide for the kind of insights, solutions, and expertise that have a worth all their own.